I'm Nikhil Mittal. I'm a graduate student in the Voldman lab. Today I'm going to talk about a device developed by two former students in our lab, which is called the BioFlip chip. And this device enables the delivery of cells on a substrate of the experimenter's choice in a configuration of the experimenter's choice. So the device itself, we have one of them here, is very simple. It basically consists of tiny wells in a piece of silicone. Each well is usually designed to just hold a single cell, but if required, we can of course make them larger. The fabrication of the device is made so that the wells are in the same arrangement that cells will eventually end up in on the substrate. So the device itself is made using soft lithography. So in particular, we use PDMS, which is polydimethyl siloxane, and mold that off of a silicon wafer. We have one of them here. So the mold wafer itself is made using standard photolithography to pattern an SU8 photoresist, which is spun onto the silicon wafer. So in a nutshell, cells are transferred onto the substrate of choice by basically filling the wells up on the BioFlip chip with these cells. We then apply the chip to the substrate of choice, often a tissue culture polystyrene. And then finally, we flip the combination over, allowing cells to fall out of the wells and onto the substrate.
I'm Flavin. I'm an undergraduate researcher here in the Voldman lab, and today I'll be demonstrating how to use the BioFlip chip to pattern cells. The day before we want to do a patterning experiment, we'll plasma bond